Good morning and welcome to our Gaia Science webinar series, The Arts of Cell Culture Techniques and Media Selection. My name is Vicky from Gaia Science Life Science team and I will be the moderator for today's webinar. Before we start, we would like to introduce our today's webinar organizer, Gaia Science Malaysia. Gaia Science Malaysia is a professional team with a collective and vast experience in the scientific industry. With more than 12 years of experience, we are one of the leading suppliers of scientific and laboratory equipment to universities, research institutions, and also industry. With more than 10 technical specialists, we are recognized as a one-stop solution for life science research with a wide product range, such as animal research, cell biology, microbiology, and plant biology. In a bigger picture, Gaia Science Malaysia belongs to a group of companies under Gaia One, which unite offices in Singapore, Indonesia, Myanmar, and also China. With a strong network across the region, bridging state-of-the-art laboratory products to the scientific community is our aim, while providing service excellence is our goal. We see the scientific community as our partners in promoting scientific and technological advancements for a better world and better quality of life for all. Ladies and gentlemen, I would like to take this opportunity to thank all of our participants for your interest and support to join us today. It is indeed a pleasure to have you all today. Before we get started, just a little housekeeping for today's webinar. Today's webinar is scheduled to last 1.5 hours, including Q&A. This webinar will be recorded. The recording and the slide will be shared upon request. We will keep your microphone muted during the webinar to avoid background noise during the session. Please submit your questions and comments through the chat box, which is visible in the Microsoft Teams tab on the top right corner. Be sure to attend the webinar until the end, as we have an in-webinar online exclusive offer. E-certificate will be provided upon completion of feedback form. Today, we are delighted to have Dr. Tan Jason and Ms. Harper Lee as our honored speaker. Dr. Tan Jason is a business development executive for Life Science Consumables and Reagents, a subdivision under Life Science team. He has vast experience in various disciplines in biology and chemistry, with his main focus on cell and molecular biology. And he is patient in bridging the cutting-edge research and providing unique solutions to his customers' needs. Dr. Tan Jason completed his Bachelor of Science from Australian National University, majoring in chemistry and molecular biology in 2013, and his PhD in biomedical science from Monash University, Malaysia in 2019. He presented part of his research on neuronal cell death pathway induced by zinc oxide nanoparticles during the International Postgraduate Research Award Seminar 2014 and also in Monash Science Symposium 2016. He has also been a judge for Young Inventors Challenge 2018 and for the Electrical and Computer System Engineering Final Year Project for 3 semesters in School of Engineering in Monash University, Malaysia. While for Ms. Harper, she is actually a senior sales channel specialist at GeneDirect, responsible for international business. She served as a frontline sales specialist, coordinating with a technical team, and providing comprehensive customer service. Harper Lee has extensive experience in different life science fields. She holds a bachelor degree in nutrition and master degree in biology for cancer therapy and microbial research, and has been involved in mesenchymal stem cell and pluripotent stem cell biomaterial engineering research. Without further ado, we would like to invite our speaker, Dr. Tan Jae Sen, to give his presentation. Dr. Tan, the floor is yours.
All right. Can you hear me, Vicky?
Yeah, it's clear.
That's good. All right. Thank you so much for joining today's call. Okay, today's webinar. Let me just share my screen right now before we start. Okay, can you see my screen?
Yes, we can.
Yes, okay. Just want to check whether we can see. Okay. All right. So, good morning, everybody. Okay, and thank you so much for sparing your time in between your busy schedules, wherever you are, even if you are a cell culturist yourself already, right? So I know that you would have a certain time points of treatments to do, okay, to come for this call, this webinar, okay? So today I would like to present to you the art of cell culture, right? So cell culture technique and media selection, right? So we got a lot of things in store for you today. All right, so this is the agenda that we have today, and basically it will cover a few things. So about what is cell culture, and a little bit on biological safety levels as well. Okay, some equipment, apparatus, and consumables that is part of cell culture. Okay, and obviously the certain accepted techniques and some best practices as well to avoid types of contamination. Okay, and then after that, we would talk about a bit on the cell morphologies, the types and the systems that exist out there, a couple of workflows, and also the environment that the cell should be in. Okay, and then after that, this is where Harper will come in a bit to explain a bit more about the culture medias. Okay, and finally, we will end today's session with applications of cell culture. Okay, so to begin with, what is cell culture? Right. Okay. So for some of you that this is the first time hearing, basically it's a process for growing cells and tissues under controlled conditions in the lab. Okay. Sometimes you cannot really do it in an in vivo model. You can actually replicate it more in vivo, in vitro, and in lab conditions. Yeah. And the idea here also is to basically remove cells from animal or plant and then grow them into a favorable and most importantly artificial environment. Okay. Now, the idea here also is to create model systems, okay, and to help you to study like basic cell biology, you're able to replicate disease mechanisms, right, consistently, and also you want to investigate toxicity of novel drug compounds as well, okay. Now, you can actually use cell culture to easily manipulate genes and molecular pathways, all right, to develop clonal and homogeneous cell populations, okay, for specific cell types as well. Okay, and it's actually very ideal for reproducing and consistently reproducing it actually, okay, compared to in vivo, right? With in vivo, right, you may have other factors, right? You know, like not putting the example rodents or zebrafish in a proper conditions, right? And that could affect the cells inside the cell, inside the organism. However, if you have it outside, right, in a controlled environment, artificial environment, right, you're able to control it to whatever research you're doing, okay? And obviously, with any cell culture, okay, it is performed in a very restricted environment, okay, and normally with proper training and basically full proficiency with good laboratory practice, okay? So now, cell culture is normally done, okay, in a biological safety level conditions, okay. So understand here that cell cultures, when you're working with cell cultures, right, you're actually working in a hazardous or sterile and highly regulated environment. Yeah. So this means that it requires your discipline and also your responsibility when handling cell culture. Okay. So those of you that are working in cell culture right now, right, okay, or those you're going to go into it, right, take it as a privilege, because not many general public, right, will have access to work with cells, right? Maybe not in their lifetimes as well. Okay. Now, obviously, you can see that there are 4 types of biosafety levels here: 1, 2, 3, and 4. Okay, and basically, these all biosafety levels, okay, which would determine how your lab would be. Okay, mean like the environment, the equipment, the processes. All right. Okay, how they are. Okay, determined by Centers for Disease Control and Patient Prevention (CDC) in the US. Okay. Um, Basically, the levels will be determined on the organisms that you're working with or at any given time and any given lab. So it means that if you ever collaborate with other researchers, not just in Malaysia, but even in overseas, like in Singapore, Indonesia, okay, or even in Australia as well, right? They would follow the same biosafety level and also all of the SOPs and protocols and safeties in place. Yep. Okay, so it's quite standardized across the board, and it's actually helped to safeguard and protect lab staff, okay, because they are the ones that are directly exposed to the organisms and also the cells, yeah. So, and it sets a certain expected lab practices as well, okay, for current and even those incoming as well, right? Because obviously there would be younger researchers who have never done cell culture before, so it's very important that they practice the same ones that those, like for myself and for others of you in the call in the webinar right now, that have done probably more than 5 or 10 years as well, okay, in cell culture. Yeah, so obviously each of these levels will build upon each other. So like BSL 3, for example, follows everything from BSL 2 and more, and BSL 4, okay, follows whatever that is required in BSL 3 and more, okay. And basically, in terms of cell culture wise, most of the cell culture facilities, right, would follow BSL 2 mainly, with rare occasions to BSL 3. Just to give you an idea, cell cultures are normally done in BSL 2 conditions, whereby COVID-19 is done with BSL 3. Okay, so you can see that we are actually one step lower only from performing COVID-19 related work. Okay, so that's how much the you can say the level is, okay, in terms of how serious that we should take cell culture. Okay, now obviously you would need the right equipment and apparatus and consumables as well, okay, to have a smooth sailing cell culture experiments, yeah, okay, or your research. So there are a couple of equipment apparatuses that you have, and all of them actually have important roles to support each other. Yeah. So the main place that you normally be working with for the most of your time, if you're doing mainly cell culture, is laminar flow hood or biosafety cabinets. Yeah. This is where anything that requires to be sterile is done in there. Okay. Then obviously with your cells, you would actually grow them, okay, into an incubator. All right. And it's normally conditioned in a certain temperature, and also the right amount of CO2, carbon dioxide amount as well, okay? We'll go through that a little bit more deeper later on, yeah? Now, obviously, with the cells you grow, you need to observe how they look like. You need to see whether there are any changes as they go, okay? With your 3 days, 7 days, 10 days, or longer kind of incubation, okay? So this is where inverted microscope will come in as well, okay? Now, obviously, once you see the cells are already confluent, okay, means that it's already filled the area, okay, of your flask or your media, then you would use a centrifuge to spin it down so that you can collect the cells, okay, or you want to do like additional subculturing, for example, okay. So centrifuge is important. You also need to know the right speed as well, okay. Now, obviously, water bath is important so that your medias, okay, or your buffer solutions need to be at the same temperature as your cells, so that you allow the exchange to be not as shocking to the cells. Okay, so obviously, you and me, we like to have things as is. We don't want sudden changes. Same for cells as well. Okay, now we also will need fridge and freezers. Well, firstly, fridge is to keep your buffers in a lower temperature, because you would have certain supplements that are temperature sensitive as well. Okay, and freezers is to basically keep your some of your cells that you put away for longer term, passages basically. Okay, in some cases you might need liquid nitrogen storage as well. So this is where you need to keep like 6 months, 1 year, 2 years, for example. Okay, now obviously before you do any of this storing, okay, or cryopreservation they call it, okay, you would tend to count your cells first by using a hemocytometer. All right, to see how many you have in there and whether it's adequate to do subsequent experiments. Okay, now with all that in mind, right, you need the right pipette as well. Okay, and obviously you need the right pipetting technique as well for that. Okay, so it's for basically transferring the cells in between flask and also to seed it in your 96 well plate or your flask or the dish. Okay, now obviously with all this in mind, most of the time where your cells will normally grow is in cell culture vessels. So you can see the flask, the dish, and the different types of plate, no matter whether it's clear, white, or black plates. Okay, and obviously you will need the right consumables as well. You know, the centrifuge tubes, microcentrifuge tubes, cryovalves, and even physiological pipettes as well. Depending on whether you are working with T75 flask all the time, means the bigger flask, right, or you're working with the small T25 flask. Okay, now you might see that, okay, there's a lot of equipment apparatus here, consumables, right? So if you have all these, you think you're good to go, right? Not exactly. So even though you might have all of these items right here, right, if you don't have the right technique, the right attitudes, and even you know, the right precautions as well, you know, then it's all kind of wasteful. Okay, so this is where I go to the part where I talk about the certain basic aseptic techniques and best practices that you need. Okay, and we'll be covering some of the areas such as like in work area, okay, in your biosafety cabinet, some of your personal hygiene and safety as well. Okay, certain handling ways you should handle your reagents and consumables, a couple of pipetting techniques you should be mindful about, and lastly, between your co-workers, basically. Okay, so let's start with the work area. Firstly, your work area must be very spacious, okay, and must be also strategically located as well. Okay, so basically you want to put in the area where it's far away from movement and traffic, because the last thing you do is that you have somebody that's from another lab, right, coming in, and then is near enough to the cabinet that something could go in, right, okay, without you knowing. Okay, so this is ideal condition, of course, okay, to have a very spacious area so that all of you, and also strategically located so that like incubator and also your biosafety cabinet is beside each other, so that you can quickly, you know, take out your flask and straight away do your disassociation, okay, or any work that you relate to cell culture. Okay, so now in terms of your biosafety cabinet, okay, it should be properly set up, organized, and arranged, okay, obviously with the necessary item that you need. Okay, so as you can see right here, all right, you would tend to have your clean stuff on the left and dirty stuff on the right. So the idea is that you work from clean to the left to dirty on the right. Okay, right-handers would normally be this, left-hander is flipped. Okay, so it depends on you. Yeah, normally what you will put is that you will put your sterile items on the left, okay, and then you will put the tubes, okay, and your flask, okay, that you'll be working with around the middle. You start to put some of your pipettes also around the middle towards the right. Sometimes it's on the left side depending on your lab setup, and anything that's wasteful you want to throw, that's dirty, okay, all right, would be kept on the right. Sometimes you will put it with biosafety have the bag there, and you might have also a waste bottle there as well. Okay, so one example here you can see is how they set up right here quite nicely. You see you have everything sterile on the left towards the middle, and after you start to have waste on the right side as well. Sometimes you might have a vacuum pump, you will put it to the right side as well, and if it have a plug, it will be connected to that as well. Okay, so the idea here is, and also you must make sure you keep the spacing in the middle as wide as possible, because like you see her, this is where you'll be doing most of your work. Okay, and this is where also like chances of getting stuff on it as well, or contamination on it as well, could be high. Okay. All right. So next one. Now this is very important. Okay, and basically it's all about sterilization. Yeah, or making sure that whatever goes in and out is clean. So normally, first thing you would do before you start and after you finish as well, you make sure that you wiped your work surface with ethanol, okay, or isopropyl alcohol. All right. Normally 70% is good enough. Okay. Before and after. So like over here, you see she's wiping the base, she's wiping the top, she's wiping the sides as well. It's good practice to actually just wipe everywhere before you start. Okay. And normally the rule is that anything goes in, even some items that you might have taken out and then you put it back in again, okay, must be sprayed and wiped with 70% ethanol. No excuses here. Okay. And lastly, when in doubt, so for example, if let's say your lecturer suddenly comes in the middle, or you got colleagues or others come in, and then you might be holding something and you be like discussing a little bit, a minute or two to arrange some timetable, and you forgot whether, okay, has it been sprayed or not. When in doubt, just spray it. Okay, there's no harm spraying additional ethanol, okay, because it's actually much more affordable than actually changing new media or changing new plate or wasting unwanted time to figure out where the contamination is coming from. Okay, so just make sure that before anything goes in, spray with ethanol. Okay, now obviously that ethanol spraying or sterilization is during your work, but before and after your experiments, right, or even as you are performing your cell culture work, right, in the subsequent weeks, best is actually to routinely clean and sterilize your incubators, which is basically any equipment that you are using. So your lab area that you're using, routinely clean. Okay, perhaps together with your colleagues as well. Okay, to clean every single area that you normally would touch, with your cells. Okay, incubators as well, you should decontaminate whenever you can. All right, spray whenever you can. Routinely do it so that you avoid any contamination as well. And if you reach a point where even you do any of the cleaning, okay, and you still have decontamination, and you still have contamination as well, consider to decontaminate your entire lab. So like the guys over here, which is our service team from GuySense, by the way, okay, is to decontaminate the entire room with hydrogen peroxide. So this is basically one of the things that you would normally do just to make sure that despite you using ethanol and doing whatever you could, right, extra precaution basically. Okay. Now let's talk a little bit about personal hygiene and safety here. Okay, so the most common thing that you could do, okay, is wash your hands regularly before and after work. Okay, because the only thing that is going to be closest to your cells and to any of your consumables is your hands. Okay, obviously be wearing gloves, but you also want to make sure that you wash it before and especially after, because sometimes, right, your gloves might break. Okay, you might be wearing those gloves, very thin latex or nitrile gloves that is quite stretchy, or might be different sizes that you wear until you got holes. The last thing you want to do is that it goes onto your hands. Okay, so you want to actually make sure that you wash your hands after that. Yeah. Now, other than that, you should be wearing all proper personal protective equipment as well. So like gloves, lab coat, and goggles as well, whenever you're present in the lab. Okay, so even the simplest thing as writing your lab report as well. Okay, you would, as long as you're in the lab, you need to make sure you wear lab coat, goggles if you don't have spectacles. Okay, and also gloves as well. Okay, because you never know where and what you'll be catching. Okay, so a good rule here would be how she is wearing, okay, or how she looks like. So closed toe shoes, long pants, no shorts, no skirts, yeah, okay, and also lab coats and gloves. Okay, now for the hair, okay, basically for that one, depends on your lab, you might need to be wearing hair nets. Okay, if not, then just like this is fine as well. If you have long hair, whether guy or girl, best is to tie it and then you put it behind your lab coat, inside lab coat as well. Okay. Now, this for sterile reagent and consumables, right? Okay. you want to make sure that basically any reagents, media, and solutions that you're planning to use, okay, you need to sterilize it, okay? Sterilize them before you start or use them in your experiments, okay? And normally you would use them only in the biosafety cabinet after you wipe it, okay? Anything that is sterilized, open them only inside the biosafety cabinet, not outside, okay? Inside only, yeah? And basically, you need to make sure you kept all your bottles, flasks, and other containers, even for those plates, you cover them when you're not using it. Okay, so like if you're halfway trying to get the cells ready and you're trying to set up right whatever around your bench in your biosafety cabinet, the bench area there, okay, make sure if you're not using it, cover it, keep it closed. Okay, now in terms of trying to aspirate the media, dispense the media, right? So two ways you could do it. Firstly, if let's say you're opening it from a flask or you're opening it from a bottle, you could hold the caps like how this person's doing, okay? Or between your thumb and fingers, or even on your pinky as well. Yeah. Okay. You can hold the caps on your hand. Okay. Or let's say if you are quite fatigued, right? Or maybe it's a long day and then you don't want to be doing that, or you're not used to it. You could do like the pictures on the right here. Okay. You could place the caps or covers, okay, down on the inside the biosafety cabinet floor itself. Now, whether open up or close down really depends on you, okay? Because you can say, if you put it open, stuff might fall into it. If you close it, the bench itself, right, might be dirty, okay? So most importantly is to make sure that the area that you're going to put in is clean. That's the easiest advice I can give you. I've done both methods myself, and so far so good, okay? Now, the last thing I will mention to you is basically, right, whatever that you open, never leave it open long enough inside there. Because the last thing you want to do is that you leave it open just for a minute or two, right? And then next thing you know, there's contamination there already, okay? Even leaving it open for 30 seconds, right? Or even 10 seconds, right? You might have a risk of stuff going into it. That's because, right, if you ever worked in the among the biosafety cabinet, there's always airflow. So the airflow sometimes could be interrupted briefly with you coming in and out, and that alone is enough, okay, to bring something in. And if you have plates, medias, or even flasks that's open, it might go in. Okay, so basically, when you don't need it, close it. When you need it, quickly use it and close it back. Okay. All right. So moving on along these lines, okay, you would have some items that are basically sterile, okay? So they need to be sterile, like your tips, your tubes, and your flask, plates, flasks as well, okay? So basically only use any items, okay, that has an autoclave tip. So like tips, and for tips, right, because most likely you'll be purchasing in bulk. So normally you need to sterile it before, but you need to basically autoclave it, right, before you actually use it. So make sure that when you are using those with the autoclave tape that's been darkened. Okay, so especially chips and plasticware, when you purchase it, it's sterile. You're going to make sure that they are basically wiped down before you bring in. Yeah. So anything you keep in resealable bags, tips, box, or glass jar, make sure they are kept sterile all times as well. And when you bring it in as well, make sure you wipe it with ethanol. Okay. Now, what you should do, right? Once the item leaves from the bags, the box, or jar. So in any case, sometimes you might take out a tip, might take out a tube, you might take out a flask by accident. Okay. So then it's considered possible to be contamination. Okay. So you cannot place it back already once among the sterile items. Okay. And if you take it out or it comes out by accident, you either use it or you discard it. Okay, so you cannot put it back together. Okay. Now, also in terms of the reagents, please routinely check before using it. Okay, so like you can do like a pre-check. You have a list to check. Okay, is my media clean? Is my buffer clean? Is my items are all sterile or not? Okay, to avoid potable contamination. This is in terms of the reagents. Yeah. So if you see the reagents or solutions that look a bit weird, like cloudy, or maybe there's floating particles, for example. Maybe you did not filter it properly, you know, or there's sometimes foul smell. This is quite extreme already, okay? Or unusual color as well. Please don't use it, discard it, okay? Not worth the risk, yeah? Okay? Even though they might look a little bit off and they might not look contaminated, please discard them and assume that they are decontaminated. If they're contaminated, so just discard them, okay? And then obviously, if the color change is very drastic, also discard them as well. You can see some examples over here. Okay, normally your culture media should be around the right side here, where it's basically neutral pH. If it goes towards a little bit alkaline as well sometimes. If it go towards the yellow side here, is considered acidic, and basically this could mean either you know, there's no nutrients in there, or there's already contamination in there. So if you see your reagents are like that, please just discard it. Okay, don't leave it there. All right, because you might also cross-contaminate your culture media with other culture media of your own and others in the fridge. Okay, and you don't want that to happen. Yeah. Okay, so let's move on. Now, couple of things here on terms of pipetting techniques as well. Okay, that this is not restricted to tips, but also to physiological pipettes as well. Yeah. So most importantly is that basically the underlying here is be conscious. Okay, so when you are doing pipetting, right, focus, because that's the time where you are actually touching the cells. Okay, that's probably the only time they're really touching the cells. Okay, so you be conscious where your tips and pipettes are touching or where they are present. Okay, also in terms of how you're aspirating it. Are you doing it too fast? You're doing it too slow? Okay, you're doing it with bubbles? Okay, so that could also affect the cells as well. Yeah. Now, one of the things you would see over here, right, on the diagram here, is to avoid touching the tips, okay, on pipettes at the mouth of the body. So you can see right here the way they're doing it. They're not touching it along the mouths, okay, or even the sides. They're going straight into the solution, okay. So you want to avoid any possible picking up items, because sometimes, right, the mouth could be contaminated. You never know, or it could have some things that you cannot see, you know, even under the microscope, even with your eyes. So you want to avoid that as much as possible. Okay. So now with regards to the one on the right here, sometimes, right, you might be doing a lot of treatments and a lot of dilutions. Okay. So rather than actually holding the microcentrifuge tubes, either 1.5 or 2 mils on your hand, consider using a microcentrifuge rack. Okay, so you can easily organize it and makes your pipetting like this easier as well. Because the last thing you do is that you hold it on your hand on the left for the tube, you hold your pipette on the right, and then after that you would try to, as if like, you know, do like a key into a lock kind of situation. The last thing you do is to touch along the walls, and you might pick up something that is not meant to be there. Okay. Now, in terms of pipette tips, okay, if you can see on the left here, you take one, not with your hand, but with a pipette. So what you do is that you will use a pipette, okay, single channel ones, okay, or sometimes multi-channel. You try to push it down, okay, as much as possible, okay, until you feel that there's enough grip and you pull it out, okay, rather than actually using your hand and trying to jam it with your hand and your pipette, okay, all right. Normally, when the pipette tips are used, use it once, and then after that, discard it when you don't need them. Okay. For physiological pipettes, it's a bit more different. Most of the time, they will come in a sealable, sterile package, as you can see over here. And what you do is that you will acquire it from the sterile wrapping directly, okay, using an aliquoting pipette right here. And then you do your experiment, and when you're done, put it back inside. Okay, make it easier, and also you will make less mess as well. Okay. Now, that being said, whenever you feel like the tips or the pipettes or even the tubes as well, okay, you doubt that, okay, is it clean or not? Did it touch the, you know, the bench or not? Right. Okay. Or you don't know where it went to. If you don't know where it went to, okay, discard it. Use a new one. Okay. Use a new one. Yep. And I'll mention lastly is that regarding the pipettes, right, over here, make sure that you are able to pipette it upright. Because if you pipette it at the corner, sometimes you would also reduce issues as well, okay? With accuracy in that sense, yeah? Okay. Next, okay, so the last one is basically the fact that you would normally work with colleagues around you. Okay, so unless you are the very few lucky ones out there to have your own lab to yourself, right, then okay, congratulations. But most of us, even myself as well, we had to share with different colleagues as well, even not just the benches, but even biosafety cabinets as well. Okay, so normally they might be working on other things, might not be on cell culture. They might be working on microbial work, they might be working on protein work, you know, PCR and so on. So they might be crossing over your area of cell culture, right? So normally best is to be present and be mindful wherever you are. Okay, especially if you're with cell culture, last thing you do is you have something from their end coming to your side, and especially, you know, for you having your cells maybe interfering with their work as well. Okay, do take your time and plan ahead with contingencies before beginning your work. So here what it means is that you know, think about what you're going to do for your 2 hours work. So like, okay, I'm going to UV all of my items. Okay, I'm also going to subculture. I'm going to count my cells using Haber centimeter, so I will need the inverted microscope. Okay, making sure that someone else is not hogging it for the whole 2 hours because they have some imaging to do. Okay, also make sure that you have access to vacuum palm, right? And then no obstruction, and then you're able to clean it down. And just in case there's anything happen in between, you need media, you're able to access. So all this, you need to think about it, because sometimes, right, your colleagues might be coming in and also need to use the same equipment as you do. Okay, now obviously, normally you'll be sharing your area with your practitioners and sparring colleagues, because obviously you and your colleague is also pursuing PhD as well. Okay, so, and especially those that is working with non-culture related projects, right? So there will be bound to have cross-contamination and accidents. Okay, so you want to avoid that as much as possible. So be alert and understand where you're working with. So you can see right here, right, this is one example where everybody had your own bench, but they're so close to each other that even a whiff of air, some items, some of the pathogens or cells from him might grow over to there, you know, or even media as well. Okay, all right. Now, obviously, when you're sharing with your lab mates, okay, be considerate, okay, and basically discuss and consult, okay, with your colleagues, because sometimes, right, you might have a team of 6 or 8 of you, okay, but you only have 2 biosafety cabinets. So obviously, you all cannot be doing it at the same time, especially if you're doing multiple treatments and multiple time points within 24 hours. Okay, so you would need to discuss and then after that plan out with them. Okay, so because your work or their work might interfere with your or their timing, experiments, and treatment. The last thing you want to have is that, okay, they are doing it and then they interrupt your timing and then you're off by an hour or half an hour, and that could affect your results. Yeah, okay. Now, obviously, have a timetable and a gentleman's agreement among your peers. Okay, so this is basically bound to you and your colleagues on who should do, when should do, and then how, okay? And also, like, you know, be considerate as well. So last thing you want to do is that you put your items in here and then you don't know who's using it, okay? Or you might be this guy waiting for him, okay, to quickly finish it because your cells are ready to go. So you don't want to be the guy with the blue you call PPE, right? Like struggling, right, and rushing because the guy here wearing red, right, is waiting, staring at you, says, "Hey, it's my turn now," you know. Okay, so that's the last thing you want to do. Yeah. Now, that being said, right, all the things I mentioned here until now, right, it's for one main reason: prevention. Prevention from what? Contamination. Not just contamination, but time wasted as well with acquiring new media and reagents because your media and reagents was contaminated because you did not do the proper techniques, okay, pipetting techniques, or you did not close well enough your media or your plates, okay, or you didn't even spray ethanol, okay, that's probably the sin, okay, or the worst case, yeah. And then you need to waste time because the cells is contaminated. You need to revive new cells, and then after that, because it comes straight from the freezers, it will take time for them to confluence. So you might waste 2 weeks waiting around for it to grow, and then after you need to subculture them and grow into more flasks because you need to do multiple plates. So that alone you wasted 3 months, 3 weeks because of some small mistake that you did. Okay. Also, because of that, right, you might need to sterilize and decontaminate your equipment as well, your apparatus or your biosafety cabinets as well. And that also would waste additional time as well that you could use, to do other treatments or even to plan ahead. Yeah. Now, that also being said, okay, even though you've done all the prevention measures, contamination could still happen. Okay, no matter whatever you do. So if you are one of those that do your cleaning, regular cleaning, regular spraying ethanol, right? Contamination can still happen. It's one of those things where you will come across in your cell culture work as well. Okay, so if you still come across that, we need to understand and know what kind of contaminations you would come across. Okay, so I'll explain a few here for you. Yeah, so the first one that you might come across is chemical related. For example, so you might have media, serum, or water impurities that might not be as pure as you thought it would be, or it might, or when they manufacture it, there's already like contamination in there. Okay, there is also possible endotoxins in there or plasticizers. So these are coming from manufacturing of the product itself. Okay, sometimes the layer that you put in, right, gets eroded and then combined with the media. Okay, sometimes there's also detergent and disinfectants that you use. It might be not to the correct ratio and it might actually stick there. Okay, and then it goes into your calcium media, right, or your buffer solutions and cause problems. So when you are reusing your glass bottle just for PBS, for example, you're making it from scratch, right? Maybe your detergent you use, you didn't wash it properly. It carries forward into the buffer solution and it reflects on your cells. So that could happen as well. Yeah. Now, obviously, one of the most common ones would be that you always come across is with biological contaminants. Okay, so you could have bacteria, molds, yeast, viruses, mycoplasma contamination, or even worse is cross-contamination of other cell lines as well. Okay, so this is normally the ones that you will normally come across. Okay, now there are other subtle ones as well, like fluorescent lighting as well. Okay, so that would cause photoactivation of the cells to act very weirdly. Temperature changes as well. So sometimes if your incubator goes haywire, or worst case still, there was a power failure, and then your fridge that's supposed to keep it in -80 or -192 failed, right? Okay, then you would have problems with your subsequent purchases as well. Okay. Other than that, there could be possible radiation and irradiation, could be from UV or could be from other certain light sources as well, depending on what kind of equipment you use. Okay, and vibration. This is more from self-inflicting, means that like when you carry, you accidentally knock on the bench, then some of the cells get lifted up by force, or the incubator that you're using, the motor, right, is vibrating the entire incubator, okay, it's like a massage chair for cells. So that also would cause problems mechanically to the cells as well. Okay, so these are the few basically big categories of different contamination you can come across. All right, so now with regards to biological contaminants, okay, you could eliminate them with antibiotics. And I think normally you would see, if you're working with cell cultures now, right, or you're planning to, your researchers or supervisors would say, okay, let's use pen strep, okay, penicillin, streptomycin, or let's use anti-mytosin, okay, or antimitotic ones, okay. Yes, I've used that myself as well for pen strep. However, please take note of a few things as well, okay. I personally have used the pen strep, right, as the last resort because I was actually performing the biosafety, I was using a biosafety cabinet and was sharing with other colleagues that actually were doing some microbial work. So I realized that was like I had to use pen strep to do so. Okay, so normally all these, right, tend to be used as a last resort unless it's already been applied in the protocol prior you coming into cell culture work. Okay, and the thing is that when you use antibiotics, right, you encourage the development of anti-resistant strains. Okay, so maybe initially you got some bacteria in there, you use antibiotics in there, but after a while, then it becomes resistant. Okay, so that might cause the problem down the road where suddenly, right, initially was low level contamination and then become full scale, because after a couple of rounds of the bacteria proliferating, right? If I want in one particular round, right, you're able to able to break through the suppression caused by the antibiotics. Okay. Other than that, right, sometimes antibiotics might actually hide other cryptic contamination, all right, and also microplasma as well. Okay, so you might be actually suppressing some of the common ones, but you might be missing out other ones that normally hard to detect. Okay, and in some rare cases, antibiotics could actually interfere with cellular processes, okay, that you're trying to investigate. All right, so do take in mind when you're trying to use antibiotics for biological contaminants mainly. Yeah. Okay, so let me go through some of the biological contaminants that you will go through, because you need to also make sure that you identify it, right? So no matter whether you have very good aseptic techniques, if you don't even know why your cells is dying, then there's no point. Okay, so this section here would be help you to identify biologically wise to see what kind of contamination you might be having, right? And at least like you get an idea on how to do it from there. Okay, so first one is bacteria, right? So normally if you look it under the microscope, you can see, okay, between A, B, and C, right? Okay, with C having full-blown contamination and all cells are dying. With B, okay, you can see some of the spacing here is a bit more cloudy than usual. Okay, so if you put it under the microscope, right, you can actually see the shape very clearly. Yeah, in terms of quick viewing from the incubator, okay, your flask, okay, should be turbid to identify as contamination. Okay, so like the one B here is contaminated, A is still okay. All right, so this one is a media without phenol red. Now, but most of the time you would use media with phenol red. So if you do so, anything that looks beyond red, okay, more a bit like towards orange to yellowish, okay, yellowish is very bad actually, but if you see like red but turbid as well, then it's considered contaminated already. Okay, and you can always confirm that using a microscope as well. Okay, and normally the change here is because that the bacteria modify the pH, so it goes from phenol red to yellow, so it makes it from neutral to acidic. Okay, now another example here. Okay, this is very obvious here. A is when you have the cells without much contamination, and B is full blown. So you can see that it's not just yellow, but you can see that it's like precipitation on there. Okay, so that means that there is bacterial growth over there, or like cells already died and it's floating in the media. Okay, so you can see if you look through the microscope as well, A is healthy HEK293 cells, and the B is basically no cells, all bacteria only. Okay, rod-shaped bacteria. Okay. All right. Next type of biological contaminant is fungal or yeast. Okay, and it's very obvious by seeing like they looks like white pebbles, and it's like connected and it's budding out, okay, from each other. Yeah. So all these ones here, these are Cho-K1 cells, the bigger ones, and they're all smaller, looks like white pebbles. Okay, under the microscope, they are yeast. Okay, and normally they have a very quite distinctive shape. Okay, and even at 4x magnification, you're able to observe, or 4x or 10x, you're able to see it really. Okay, obviously in your flask, okay, it will appear turbid. Okay, and normally you will only really see the color change in high contamination situations. If in low contamination, normally you need to observe it through the microscope to find it. Okay, but for yeast, because they do have a certain distinctive smell, so you can actually check, you can actually identify even before you see under the microscope. Okay, and the bad part about having fungal contamination is that it can spread. So like if you do not have proper capping, or you have multiple flasks in the biosafety cabinet, and then you might be opening a few of them, right? It could spread from one flask to another airborne. So you need to be careful about that. Yeah. So another picture over here showing that these are the cell right here, and these are all basically yeast. Yeah. Okay. So this is a picture here showing that the cell is being contaminated by two different ones. So bacteria and yeast. Okay. So you could see you know, the differences very obviously. What kind of contamination? This is obviously very bad. You have two in one. Okay, but also you need to know that at least identify what is it there. At least you know that this is not cells. okay, to begin with. Yeah. Some another example of fungal contamination as well. So again, these are still yeast. Okay. These long ones are cells, and the rest are all fungal ones. Okay, they're not bacteria. Yeah, they're fungal. Okay. And you might have even worse cases like hype. So this is all like very long, fragile like arms. Okay, and then they look like not say neurons, but they look like very long kind of wires, you know, branching out from the center. Okay, so these guys here are hype. These are cells. So you know that from here, these are fungal or more mold-related contamination. Okay. Now, another type you could come across is viruses, and these are the harder ones. As you can already see from the picture here, it's hard to identify. Okay, and you're not the only one here, and the eyes are not deceiving you. It is hard. Okay, it's not visible to light microscope at all. Okay, but when there's viruses in there, it does take over the whole cell's machinery and reproduce whatever they want. Okay, in most cases, right, because viruses are sometimes hard to come across, okay, sometimes, right, you might not see them at all because, right, they have very stringent requirements to grow. So especially they need the host, okay, and even with host also, they need certain conditions as well. So most cases, virus contamination is not as big as the others as well, okay. But let's say if you see the cells are acting weird. So for example, you can see A, D, G, and J, by the way, the one over here, they are all unaffected by viruses, okay, and the ones here B, C, E, F, H, I, and K, L, okay, they are all at least being transfected by virus. So you can see, especially with A, B, and C, there are some changes, okay. The rest are not too obvious, okay, but if you want to really confirm it, you can actually do immunostaining, antibodies, ELISA, PCR with viral primers. That's the best you can do, okay. Moving on, now we got mycoplasma, okay, and this one is one of those that is also a bacteria, but unlike normal bacteria, they're so small that sometimes you don't even know it's there, okay. It's not as bad as viruses, but it is sort of hidden because it's so small that you might think, you know, it's just something normal, yeah, okay. So it's very difficult to identify until they are present in very high densities, okay. So you can see right here, A is normal Hep2 cells, okay, but B, C, D, E, and F, they are all being contaminated by the mycoplasma, and you can see the morphologies are different from A. So you know that it's kind of weird already, okay? All right, and you can see that their behavior and their morphology has been modified as well, okay? So they can alter the behavior actually if you have mycoplasma long enough, okay? And if you have a chronic mycoplasma infection, means that if it's very overwhelming, you will see and you tend to notice the cell proliferation rate decreases. So maybe you would normally see that the cell's supposed to conflow in 3 days, now taking 2 weeks, right? And then you didn't see any bacteria, you didn't see any yeast, most likely it's not a virus. So it's possible that it could be mycoplasma, okay? Like cells still die for no reason, okay? All right. Or you can notice that there's reduced saturation, means that there are less cells that is covering the area, or even there could be clumping, agglutination is clumping, okay, in especially with suspension cells as well. Okay, so you know that it's possible as mycoplasma. Yeah. Now, the way to identify it is actually through staining. That's probably the only way you could do so because it's so small. Yeah. So basically, you could periodically stain with fluorescent staining. Okay, so you could use Horsch staining, which is normally the common one, okay, or you can do immunostaining, ELISA, PCR, or even certain assays as well. So there are kits for mycoplasma detection as well. Okay, so you can see the one on the left here, they actually stain it. You can see that outside of the cell there are all these small, you know, look like Christmas. Okay, so all of these small dots here, they are mycoplasma. Okay, so you could use mycoplasma removal agent, okay, or any commercial ones out there, okay, and if you put that in, you see, okay, after you stain it again, it's very clean. You don't see anything between the cells, okay. Another example of how mycoplasma will look like is when you have progressing contamination. So you can see this is normal cells, okay, This is with microplasma, and this is with the microplasma overwhelming the cells. So when you do the staining, you will see it's like very saturated in terms of the fluorescence signal. So you know that there's microplasma there. Okay. Now, other than that, if you have other cell lines as well, okay, you could actually have two different types, right? And that could cause cross-contamination. Okay. That's one of the other ways you could get that as well. Okay, so like in this picture here, you can see you have one type of cells, okay, primary human keratinocytes, and then you will have fibroblasts as well. So you might have two different cell lines, and that could be problematic for you. You could also, for example, right here, you have normal human fibroblasts, and then you have HeLa cells as well, round ones as well. So obviously you have two types of cells in there. Okay, so this is also another contamination as well. Not so harsh, but it obviously will impact your result your experiments down the road. Yeah. Okay. And normally happens when you have fast-growing cell lines or even HeLa cell lines that might come into your culture by accident. Yeah. Okay. So you want to avoid it by checking the characteristics, practice good aseptic techniques, and also check using fingerprinting, character analysis, and isotype analysis as well. Okay. All right. So that's all the contamination that we go through. Okay, so we come to the midpoint. We got a bit of polling here. Okay, if you can see the chart on the right side here, and I believe Vicky or Suha would be putting up the polls right now. Okay, so we just got a couple of questions. Want to identify what kind of contamination that you've faced before. So it's multiple selection here, and basically, do you want to get an idea what you've come across? I personally have come across bacteria most of the time. That's how I use pen strap. I actually have come across fungal spores as well. Then I had to discard it. Okay. I think the fungal spore came from culture media. I'm not sure why it got in, but most likely because I didn't wipe it correctly. Yeah. Okay. So I'll select my one as well. Okay. I haven't come across viruses. Okay. So let me look at the poll. I'll just, okay. So if you look at your polls, right, it looks like most of you guys, I'll give you guys a few more seconds. Okay. Cool. Looks like we got 60+ responses. Yeah, so please, it should appear in front of you or it should appear on the chat. Please select them as well, yeah, to get a good idea here. Participate as well, so that at least if you're doing it for the first time, right, at least you know, okay, you might be expecting this kind of contamination, okay? So don't feel very weird that, you're the only one that's getting. I thought I was having the issue as well when I got it many times. Apparently, it's a very common thing, okay? All right, so, okay. Well, it looks like most of you guys are bacterial and fungal related. There are a couple of viruses, which is interesting. Okay, there's also mycoplasma and cell line cross contamination, and also we got those that haven't done any cell work or luckily still haven't got yet. So congrats to you if you haven't got anything yet. Okay, all right. So let's move on. Okay, to talk about the cells. Yeah. So after knowing all of the different the labs that you're working with, equipments, and also the technique and contamination. Okay, so let's look in terms of the cells, right? So because you need to know what cells you're working at as well. Okay, all right. So there are 4 types of morphologies. First one is fibroblastic ones. So they look very long and elongated, and they are normally grown, okay, on the surface. So attached on the flask or the plates. Okay. Now, we also have epithelial-like ones. Okay, so they're a little bit more like pentagonal or hexagonal, 5 sides, 6 sides, right? So you can see they're a little bit more round, not say too round, but they have some polygonal shapes. Okay, all the names of polygonals that you learn. Again, they're grown also attached. Okay, you might have lymphoblastic-like cells. They are spherical and they are generally grown floating in your media. We call them suspension cells. Okay, and lastly, there's also one called neuronal, and this is very obvious. Unlike the fibroblastic ones, they have axons and dendrites as well. Okay, so they look very long. They could be bipolar or multipolar as well. Okay. Now, a couple of example cell lines that you might come across. Again, these are the common ones that I've seen. There are obviously hundreds of them out there. Yeah. Okay. So there's one HEK293 cell line. This looks epithelial. Okay. And they are embryonic human kidneys. Okay. We also have A509 cell line. They are a bit more epithelial-like as well. Okay. And they are basically from Chinese ovary of an adult Chinese hamster. Okay, there's also the A519 is lung carcinoma epithelial. Okay, K1 ones they look a little bit more elongated. Okay, and they are basically human cervical cancer cell lines. You have also HeLa is human cancer. Okay, K1 is more ovary of adult Chinese hamster. HeLa cell line, okay, is more human cervical cancer cell line. You can see they are irregular shaped, so these are polygonal ones compared to K1. HuVec ones, they are human endothelial cell line from umbilical cord. Again, they look like in between K1 and HeLa, right? Okay, the shapes. Okay, then we do have neural stem cells. Okay, so they look a bit more neuronal right now. Okay, we also do have C3T3L1. Okay, and they are basically mouse adipose tissue. Okay, and we do have SF9. This is insect cells, by the way. Okay, and they are lymphoblastic-like suspension. Okay, so these are like, you can see they are all different shapes and sizes. Okay, so, and even like how the width of the cells are also different as well among each other. Yep. Okay, so now in terms of cell lines you can culture, is mainly in two classes: finite and continuous. Okay, so in short, finite is basically those that has a certain lifespan, and continuous is forever. Okay, so under the finite category, you would have, and you normally hear about, primary culture or primary cell cultures. And in short, they are basically isolated directly from animal tissues or organs. Could be humans as well, actually. Okay, and they are immediately proliferated under conditions that matches with closely with the host. Okay, so once they are collected, okay, they are then subcultured and then into cell lines. So after the first passage, right, primary culture will be renamed as cell lines. Okay, and again, there's still a limited time span as well because they're directly from primary cell cultures. Okay, and but they passage with the highest growth capacity, and they have very wide range of genotype and phenotype as well. As you go down the road, right, you might be deciding to branch out a little bit to other types. So we call those cell strains as well. Okay, and they are subpopulation from the main cell line. Okay, now from all these cells here, right, you could convert them to continuous ones. Okay, and one of the two types is transformed cells. Okay, so some examples here is CHO, HeLa, and HuVec cells. Okay, and they are generated through certain transformation and genetic manipulation. Okay, so they obviously have higher growth rates. Okay, and also they require less serum as well. Okay, and obviously because they are conditioned to this particular type of media that you're planning to do. Okay, so they have higher plating efficiency as well. Okay, so if you want to do many cells plating and many treatment very quickly, you could do so. Okay, now they are obviously stable and standardized compared to the finite ones, but they might have some karyotypic anomalies. Okay, and certain phenotypes that don't exist in primary cultures. Okay, now other than transformed cells, we also have self-renewing cells, and these guys are actually more on the stem cell side. So you can see embryonic stem cells, iPSCs, neuro and intestinal stem cells. Okay, so because these self-renewing cells, they're able to divide indefinitely and make more of cells. Okay, which you can later on differentiate to other diversified cells as well. Okay, and this here all basically you're able to do long-term maintenance as well. Okay, you're able to really grow long as well. So there are basically two classes of cells that you could be coming across. Okay, now with all that in mind, you need to decide what kind of cell line do you want to use. Okay, so couple of characteristics you need to take note of, or factors you need to take note of, is the species. You know, whether it's human, non-human, non-primate, or insect. Okay, some of them you can say have less biosafety restrictions, so it means it's easier to obtain and easier to run. Yeah. Some of them we might want to look on based on their functional characteristics. Are you doing toxicity testing? Are you doing drug bioavailability? Are you doing transfection? Or maybe differentiation or specialization to specific type of diseases? For example, you know, you might use the neuronal cells to condition it by differentiation to Alzheimer's or Parkinson's disease, for example. Okay, whether you want to choose a finite cell lines or continuous, so that depends on whether you want more functions to be expressed, right? You'll be looking towards that, or you want those that you focus on particular type of expression, okay, and then you want it to be easier to clone as well. Okay, now other than that, you might be considering whether you want the cells to be just normal, means like generalized, or you want it to be transformed to a very specific type of cells. Okay, so normally if you transform, you know, you might be having basically, you know, faster growth rates and then less serum, so very good in the long run for saving costs, but it could be permanently different from the normal cells. Okay, also you might be deciding what kind of conditions and characteristics you want to subject the cells to. Okay, you know, can they grow in suspension? Can they grow on the flask itself? Okay, and also you want to choose which one can grow quickly, or whether they can even express recombinant protein for your work. Yeah. Some other criteria here you could involve, like whether you want to use finite cell line. So are you able to access rats or zebrafish all the time, or insects? Right? Maybe you only have 2 rats or 1 rat. Then you are limited. Okay, because you cannot be, you can say, harvesting the cells and then after that hope for the best. Okay, especially primary cells, you need to harvest continuously from fresh stocks. Okay, is your cell line very well well known, basically well published, or it's new that you need to perform validation by having another normal cell line that is very equivalent. Okay, it is stable to begin with. You might be, you know, just a certain vibration, your cells will die. You never know. Okay, or is it easy to be cloned and even enough to generate sufficient frozen stocks for subsequent experiments? So all these are factors that you need to take note as well. Okay. Now, there are 2 systems among all of that, right, that you would normally be coming across: adherent cultures and suspension cultures. Okay, so just to keep it short, okay, due to time as well, adherent cultures are those mainly that would be basically growing, okay, at the base, all right, of your flask, culture plates, and also dish, and they are basically limited by the surface area of where you're working with. Okay, you need a tissue culture treated vessel because they would have to be attached, okay, to the surface. All right. On the other hand, for suspension, okay, they are normally floating in the media. Sometimes we call it floating cells. Okay, you would okay, the growth wise is limited by how much area or how much medium you put in. So if I could use an analogy of like a house, okay, adherent cultures will determine how much space that you have. Suspension will determine how much height you have. Because some houses, right, are very low ceiling, or even malls. Some houses or malls have very high ceiling. So those that have very high space, right, okay, which will be represented by a lot of media, right, would be ideal for suspension culture. And again, limited by how much media or the height of the media is. Okay, all right. So unlike the adherent culture, suspension, you don't really need to have a culture vessels that's treated. Okay, but because, right, it is floating, okay, occasionally you need to come in, shake it a bit, stir it a bit so that you can move around, okay, and you allow some of the media to exchange around. Okay, so with all that in mind, we need to also know, okay, or at least get an idea what is the typical workflow as well. Okay, so basically, right, you would start from thawing your cells. So this, if you buy the cells from if you buy from like example from ATCC or Merck, right? Okay, you will thaw the cells. After that, you put it into flask to incubate it. Okay. If you're doing primary, that's for cell line. If you're doing primary cell cultures, okay, you'll be basically extracting it from your host. Then after that, you will making sure you would roll down the flask as well. And in both situations, when it reaches confluency, you will subculture them into new passages. Okay, you see them and grow enough that you're able to basically have enough to do treatment. So you continue to expand with multiple passages until you're ready to do treatment. Okay, so at the passage stage, with one of the passages that you're doing, okay, you will check the morphology first, of course, to see if there's any contamination. But also, most importantly, you check the confluency. You normally need about 80% or 90% confluency. Okay, sometimes 70%, depending again on your cells and depending on your SOPs. Okay, if it's enough, then what you do is that you will trypsinize so that you will disassociate, okay, and you will suspend the cells. Once you suspend it, you take some and you perform counting either using a hemocentimeter or an automatic cell counter, depending what you have in your lab. Okay, then once you count enough, you could do a few things. You could seed it, okay, into a well plate to do treatment. You could also seed it back in a culture flask to grow for the next passage, or you could cryopreserve it straight away as well. Okay, so for treatment-wise, you could do some study analysis as well, PCR, luminescence assays, biochemical assays like MTT, LTH, ROS, and so on. Okay, just to check what is the condition of the cell post or pre-treatment, or whichever works for you, or recovery as well. Okay, so some of the treatments you might preserve it for future. Like example, you do treatment, you preserve the cells that might have certain DNAs and RNAs, okay? Maybe not to the point of preserving it at -80, okay? But at most cases, right, when you have too many cells and you want to keep it for future, you can always cryopreserve the passage itself, okay? So that you can revive it when you have contamination, okay? So normally you might use certain freezing mediums like DMSO, glycerol, or like a dedicated like CD freezer medium over here, okay? You will actually harvest it, preserve it, and then you would subsequently freeze it subsequently, like -20 first, then -80, then put it in nitrogen for long term. Okay. Now let me talk a little bit on other techniques that you should take note of. Okay. Now for subculturing and passaging, it's basically the idea to first mainly to remove and exchange medium, but most importantly is actually to transfer cells from the current vessel, which is already too confluent or too many, to either a fresh grown medium or another culture dish, flask, or plate. Okay, so basically you want to further propagate or expand and maintain the cell lines. Yeah. Okay, so basically you want to try to keep it to a strict schedule. So like basically try to make it every 5 days, 3 days, and so on. It's like when you go to work, you have a routine. You don't want unexpected things to happen. Cells are also the same thing as well. Okay, because when you make it a bit irregular, then they also will act very irregular to your results as well. Okay. Now, you normally perform it when for adherent cells case, there's no more room left, and for suspension cells case, when there's no more room in the media. Okay, and this is based on observing in the microscope. Okay. Now, in terms of when you should do subculturing, there's two. The first one is basically based on observation. Okay, so this is where basically your subjective based on your subjective judgment, or based on experience, or even advice by your supervisor or your colleagues, right? Okay, you will determine, okay, when is a good time to do it. So normally in terms of your cell growth, right, your cell will tend to confluent the most around towards the end of the log phase. So right here, if you see my mouse here, this is where that would be about 70 to 80% as well. Okay, so when you reach this stage, right, you should subculture already, because if you leave it too long, when it goes to stationary phase, right, it might reduce the confluency of the cells and even die. And if you actually do it too early, right, your cells that might be in the lactase might be too early, that it might take 2 weeks or a week longer than a typical 1 week of confluency. Okay, so you need to be very careful on which part of the log phase that you decide to subculture. Okay, now the easier way that I personally found as well is visual, okay? Other than observing as well, okay? If you see a color change, especially with the growth media, okay, because sometimes you might be growing it longer than 3 or 7 days, because sometimes stem cells might be working with 20 days. So obviously you would have to change media in between, okay? So when you check, okay, there is a color change from basically pink to even starting to see a little bit darker, lighter red or even orange, it's probably a good time to change or subculture, because by then most of the nutrients have been used up already, okay? Or lactic acid has been produced because obviously cells will produce waste as well, okay? And obviously if it's yellow is bad, okay? But sometimes yellow also would mean that you have 100% confluency. It means there's no space at all. So this is where you need to actually make sure that you subculture it already, okay? All right, so just briefly a little bit on the adherent cells, because suspension is very straightforward. You're able to actually remove everything out from the flask and then perform the subculturing, okay? But for adherent cells, there's an additional step. We call it disassociating, okay? Sometimes layman terms we call it lifting it up from the flask itself. So basically, the workflow here is where you would perform trypsinization, okay, all right, or you might do certain things like shaking it off or scraping, okay, and then from there you would centrifuge it to spin it down, and then after that you remove old media and you resuspend it into a fresh media. Then you might do some counting to see how many you want to put it back in or how many you want to take some away to do your treatments, and then from there you would let it incubate back again, okay? So again, these are some of the procedures you could do. You could shake it off, you could scrape it, okay? Or shake it off means like you can knock it a little bit if it's very loose. But most of the time you might use, you will use enzymatic dissociation like trypsin DTA or TRIPE-RC, okay? All right, so now with all that in mind also, okay, you would need a very good environment for the cells to grow. Okay, so even if you subculture correctly, right, if all the other factors that allow, okay, the cell culture to thrive, right, okay, you know, if you did not put it in the correct environment, it might cause problems, yeah? So there are a couple of factors here you should take note of. First one is the temperature. So you're going to make sure that the cell types here that you're looking at in the correct temperature. Normally for your case it will be 37 degrees most of the time. Second is the pH level. You're going to make sure that most of your cells are in the correct pH. Most cases is neutral, slightly alkaline, okay, unless you're using insects. Third one is your CO2 levels of the incubator, okay. Normally, you would need them to be normally around 5%. I've normally seen 5%, okay. Fourth one is osmotic pressure, and this is more on like making sure that it's hydrated, because last thing you do is that you have no water, and then after that, all of the liquid, all of the, you know, the plasma from the cells get extracted out, and then you have imbalance of exchange of nutrients or waste. Okay, so having the right amount of sterile water would be autoclave sterile water at the basin in the incubator is important. The last one, okay, which is a big one, of course, is the culture medium. Okay, so basically, culture medium here, okay. It's important because it has everything. It has the nutrients, growth factors, hormones, and helps to regulate pH and osmotic pressure as well. Okay, so unlike basal salt solutions, which is like very I believe is hypotonic to the cells, means that you are pulling it out. At least the culture media helps to balance that. Okay, three types that you normally come across here is basal media, okay, which has everything that's basic, so amino acids, vitamins, inorganic salts, okay, and normally you will supplement it with serum. So this is the most basic one, okay. You may have another type after that, which is reduced serum. So this is a slightly modified formulation, okay, and this helps to also reduce the serum but produce the same results as well. Or even better, you might have a serum-free media, okay, still has also nutritional and hormonal formulations as well. Okay, but this is very specific for certain cell types as well. It probably has been validated a few times as well. Okay, so as you can see, among all five of them, cell culture media is the most important one. Okay, because think about it, you have glucose, you have inorganic salts like sodium pyruvate, okay, sodium carbonate and other things. You might have vitamins. You might have attachment factors as well. You might have serum, of course. You might have buffers as well, phenol-rate, okay. You might have amino acids as well, and even the pen strap or one of the antibiotics as well, okay. So knowing this, okay, every media is different. Every media is unique as well. So you will need to know what kind of culture media and what supplements is inside there, okay, as it is very dependent to your cells that you're working with as well. Okay, now, just to give you an idea how complicated it is, okay, you see these cell lines over here, this is a very small list, okay, of the full list that you can get from QR code. So you can take out your cameras and scan a bit now while I just mentioned to you that if you see HEK293 cells, you need MEM with 10% FBS, but 3T6 you need DMEM with 10% FBS, okay, or like BHK21, you might need GMEM or MEM or NEA, okay. So you can see that each cell line requires very specific mediums for it to grow, okay. And this is where I will pass the webinar presentation to Harper. Okay, so she'll be describing a bit more about the cell culture media. Okay, Harper, take it away. Harper, we cannot hear you. You're muted.
Hello, good morning, everyone.
Yep, we can hear you now. Go ahead.
Hi.
Yours.
Okay, thank you for Jason's awesome introduction about the cell culture. And now I'm moving forward to the next section and about the cell culture selections. So now I'm going to share my slides. So can you hear my slides? Is it work? My slides?
Yes, we can see it now. Yes.
Okay, so in previous, Jason has already introduced about the cell culture environment facility you need to have and the materials you need to prepare for the cell culture. And now I'm moving forward to how to select the cell culture media. So there are some topics I will introduce in this section about the general cell culture media selection and preparation. And during using the cell culture media, maybe you will have some concerns if you would like to change the existing cell culture media. So, and then I will briefly introduce about the Simply cell culture, about its manufacturing and quality. And then I will bring into some of our highlight products. One media is an alternative for the major used basal media, and also the products for the serum reduction purpose, which Jason also mentioned in previous for like a reduced serum formulation for the cell culture media. Mention about the cell culture media, I think most of you probably will be familiar with the name ATCC, American Type Collection Collection. Oh, sorry, I didn't remember full name. But this is kind of a dictionary of the cell culture you are using, because this organization, they provided the most diverse micro products and also cell culture products here. So if you have some cell culture, if you have some cell lines and you would like to look into what kind of conditions you need to culture with, so you can go check the ATCC, so you will get the information about the cell lines. And then you probably will be also know about the Gabico, which is the dominant brand in the cell culture market, and this is quite famous. So you will also know about the Sigma, VI, Hyclone, and stem cells. And actually now recently there are a lot of new rising brand about the cell culture, and Simply is one of them. We started this brand since 2018. So the following, I will also introduce the what Simply can serve in the cell culture media, in the cell culture supplies. So first, if you would like to do a media selection and preparation, and there are usually two ways to prepare it. So the first way is once you have the cell line, you can go check their product sheet. They show the guidance there. So they will show you the handling procedures will be like this. This is from ATCC. So it will mention that for this kind of cell line, you need to use what kind of medium and what kind of supplement. So here you can see for this kind of cell line, it needs to use medium 199, contains 1.5 gram per liter sodium bicarbonate, and also the concentration about the supplements they are using, 10% fetal bovine serum, which is quite commonly cell culture compositions, and also some essential factors needed to be added in the cell for the cell cultures. So they will mention in the product sheet. And the other way, which is also very commonly to use in the lab, is from the publication, right? Because the publication in the material methods, they will introduce the cell line they are using and the brand they are using and the concentrations and what kind of components they are adding. So through the publications, you can also gain the information about how to culture the cell line you are using. But for most of the time, maybe we are not able to have this almost the same brands from like indicated on the publications. So many of time we are thinking to seek alternatives. So when we are thinking the alternatives, maybe there are some concerns will be rise. While you would like to change the recommended brand. So by using other unfamiliar brands, maybe there will be some concerns, like will be the formulation the same as the brand that you would like to switch from, and is the quality good, and how is the performance. So the following, I will gradually solve the concerns you have about to change to another brand, especially Simply. Yeah. So this is a media formulation of the Simply. So from the formulation sheets, you can find that there are many 4 parts indicated in the formulation: amino acid, vitamins, inorganic salts, and other components. So basically, in most of the cell culture media, their amino acid and vitamins are almost not very different. The most different part will be inorganic salts and other components. For example, your cell culture media, you don't like it to have the phenol rate. So with or without phenol rate, low glucose or high glucose, or if you would like to have a sodium bicarbonate or not. So the difference about the medium formulation is mainly about in organic salts and other component parts. So for example, we pick up the 3 brands, including in the Simply brand from the cell line dictionary, ATCC, and give codes about their DMM high glucose products. So this is their name with their catalog number. We check out their formulation data sheets and do the comparison. You can find out that actually their formulation are almost the same among those brands. And probably you will find out some slightly difference, such as the sodium bicarbonate from ATCC of this product will be 1500 milligram per liter, but Gapcos and Simply's, they provide 3700 milligram per liter of the sodium bicarbonate. But actually, sodium bicarbonate is mainly for maintaining the pH of the cell culture medium in the presence of 4 to 10% carbon dioxide. So actually, if you have higher sodium bicarbonate, they will be able to tolerate under a variable pH value. Their adjusting ability will be better. So actually, the formulation almost the same we can find from brand to brand. And if you would like to thinking about changing the brands from, for example, Gapcos, we have like corresponding tables for you to looking for. So for example, you are using some products from Gapco, so you can check their catalog number and find an alternative one from Simply's. So those products are mainly very commonly used in the laboratory. So we are serving this kind of basal media as well. So by solving the formulation issues, then we will go next to the quality issue. So mentioned about the product quality, there are two important things to keep a good quality of the cell culture products. The first one is the facility we have, how we manufacture it, and the other one is what kind of raw material we are using to manufacture it. So the specs we have to manufacture our cell culture media is using a GMP compliant standards manufacture it, and we also have some ISO certificated. And the water we prepare to produce the liquid media and supplements, we using the water from water for injection system, which means this water is injectable and it's very clean and with very high quality. And the raw material we are using to manufacture, at this, we usually find the pharma grade from Merck, or if we couldn't find the pharma grade material from Merck, we will at this find from the Sigma molecular biology grade as a raw material to produce our cell culture media. So we can guarantee a good quality of the Simply culture media. And here is some facility about our one part of our factory. This is a water for injection system. And we also have our own lab to do some investigations or research and development. And here you can see that for our product development, most of we use a lot of our own cell culture media as well. So here is for you as a reference if you would like to switch on to a new brand which is cost effective and also with high quality. You can think about the Simply cell culture media. And then mention about the quality control. Jason just mentioned about the cell culture environments. They need to maintain on the to ensure the cell culture media, they are bacterial and fungal free, and with low endotoxin and with the suitable osmolarity and pH value to allow this good growth for the cells. So this is the basic quality control specs we need to do for the cell cultures, but there might be some additional demands. Depends on the company. They will further test whether there's a mycoplasma or not, or they will do SP2 toxicity test. This is just to ensure the cells can be grown under grow appropriately and healthy. So mention about the quality, and then we will think about how is its performance. As I mentioned before, we started from 2018, and it also take time for users trying to use our cell culture media. And now after 2020, there are few the users that after testing our cell culture media, they put into their researches, and then you can find some of our products in the publications, like our basal media, antibiotics, or penicillin and streptomycin supplements like L-glutamine, and also dissociation reagent like trypsin, EDTA. So from the concerns of using unfamiliar brands, then now you know the formulation. If you can check, make sure the formulation is the same as you would like to change from, and then you check you have the quality assured, and then the publications can also send credits of using this kind of product, then you can consider to do it. So after introduce then I am going to introduce about the category of the cell culture we have. So here you can see we have the basal media, supplements, antibiotics and antibiotics, cell dissociation reagents, and some benzo solutions. So, and those products is to fulfill the basic cell culture demands. But if you have some advanced demands during your cell culture or research, we can also support some extra service. For example, we have some cell culture media for the specific cells like mesenchymal stem cells, neuron cells, or hybridomas or insect cells. And if you have some spatial formulation, would like to produce your own like cell culture media, we can also do a customized liquid or powder media service for you. And then we also do some development about the advanced formulation. For example, one medium, I will introduce this product to you later. And glutago. Glutago, you can take it as stable L-glutamine to like maintain your cell culture media in a good pH value and also ensure cell can grow well under the environment. And stable L-glutamine, you may be familiar with the brand from Gibco named Glutamax. So this is the same product as Glutamax. And if you have the demands for less FPS consumption, because now the trend of doing cell culture, they are trying to using less FPS during the experiment. Probably they would like to go to a clinical or maybe the research purpose. So they will have this kind of demand to reduce the searing during their cell cultures. So we have some products to fulfill these kind of demands as well. So the following, I will briefly introduce these two kind of products we have for your reference. So while doing the cell cultures, we all know that we need to prepare the different basal media to allow cells to grow in a suitable environment by using the right basal media. And if we have the wrong media, it probably could get a poor cell growth. So during your cell culture, if once your cell culture work getting heavy, or if your cell culture condition is getting complex, then this kind of cell culture preparation might be an issue while you are doing this kind of work. So we are thinking whether there's a way to simplify the cell culture, and this is the idea why our one median comes from. So we developed this one median to fulfill as an alternative to replace the major used basal media in the lab, which means you can use one median only to culture on your cell lines and to have them also grow in a good conditions, and they can grow well. So there are some tests we have done by using our one median. So we have tried the one median on culturing different type of cells. For example, cancer cells and HAC cells or epithelial cells, T lymphoblasts or B cells, mesenchymal stem cell as well. So we have already tested a lot of cell lines. So we've already ensured this one median can sustain their cell proliferation and to have their gene stability are tested as well by using STR-PCR. This kind of analysis is to make sure the cells is the same as you have before you change the media and after you change the media, that the cells you have and cultivated under one median, they are the same cells without genetic changes. And also we test the cellular bioactivities as well. And here I would like to put more emphasis is that from many cell types we have already tested. There are mesenchymal stem cells we have also tested here. And after using the 1 million cultivation, you can we found that the cells can maintain its MSC definition after cultured. And here I would like to show some performance about using our 1 million. So the human bone marrow mesenchymal stem cell we harvest is from its kind of primary cell line. So it's finite cell line. So they have a certain lifespan. Span is very short. It's not continuous culturally, and by using this kind of cell lines, the most important thing is we need to harvest a lot of cells in the end to do the differentiation, because the stem cells characteristics is they are able to be differentiated into some certain lineages, right? So after several culturing passage, and we check their doubling time. Basically, for this kind of primary cells, their cell doubling time will getting longer after passages. So you can see under the traditional cultures you have, then their cell doubling time will rising. But by using 1 million in the certain conditions, in the same conditions with additional 20% FPS, in it, their cell doubling time maintain very similar, even cultured by few passages. So I think this is very important for the stem cell cultures. So we also check their stem cell marker, surface marker, CD90 and CD105 expressions compares to the control one and using 1 million one, then and their expression also shows positive. And here is the morphology checking. They didn't show a morphology change after culture. So, and then after culturing, we would like to check the most important part is whether they are be able to be differentiated into the 3 lineages. So we've done the test by check their adipose site differentiation or osteogenic differentiation. This is alizarin red as staining to check their calcium concentration. So here you can see by using our 1 million, they can show a good differentiation ability. And then the chondral site differentiation also we doing this. So here with our outstanding differentiation test by using 1 million culture, we can see a good performance. So after those tests, we have already ensured that 1 million is able to sustain cell proliferation and maintain their gene stability, and also remain their stable functions of the tested cells. And this median also exhibits good potential in mesenchymal stem cell cultivation. So that's why we said it will be a good alternative for the major used basal media, because it can be changed from like DMEM, DMEM12, RPMI, and so on. So so this is for a product for your reference. If you need, maybe you can check with the Jason Gaia Science for more information. And then I am moving forward to the product for serine reduction purpose. So basically in the traditional cell culture, for example, 10% FBS content complete median, it just simply prepared with traditional basal media plus 10% FBS, then it will be a complete median for the 10 FBS content culture media. So for the serine reduction purpose, we have some products to introduce you to. So we have trying to using a different way to reduce the FBS usage during the cell culture. For example, SR basal media is an alternative for the traditional basal media. So when you change your basal media into the serine reduced basal media, SR media, so you can just simply add it in very low FBS, 1 to 2% only in the cell culture, so you can reduce your serine consumptions during your cell culture time. And the second second one we are trying to modify it is on the FBS part. This kind of component is just simply mixing with the serum reduce buffer with the FBS you are using, and as a supplement to supplement it in the basal media, so you can have a complete media with only 2% FBS content in your culture media. And the third one is the supplement, to just simply add it into the media you have and just reduce the FBS you are using. So this kind of cell culture media is able to be to allow the cell growth in a healthy way, and the same as the original 10% FBS content cell culture media. So here, the following, I will show the performance after using this kind of products. For SR basal media, we've already compared with the similar product with Gibco's, and its recommended concentration is using 2% FBS, and we test our SR basal media with only 1% FBS or 2% FBS here. So the morphology shows no different changes after cultivation, and then also check their cell doubling time. And it's quite normal if we change from a traditional media into a new media. So here, basically, cells they will need the time to adjust the environment changing. So you can see the cell doubling time will rising, but after they adjusted the environments, their cell doubling time will go normally. And here, I would like to further pick up that. By using our SR basal media with 1 FBS supplemented, you can see the cell doubling time, although they need some adjusted time, but it seems like their adjust time is shorter than the Gibco ones. And also, if you supplement it with 2% FBS, then their growth rate is almost no difference. So this kind of products, they can have an 100% pain-free during the transitions. You need cells; they need less time for the adjusting, and you can also save most of the FBS, about 90% FBS reduction in the formula, and also only 1% FBS remained in the media. And we also do an STR verified by using our product to ensure the cells we have is the same as the control one. So the next product I'd like to introduce is the SR buffer. It's just aiding with the FBS you have to make it as a working solution SR supplement to grow the cell. So you can see their doubling time is normally and similar as before changing. And we also do a cell function by expressing the GFP proteins by transfection using 2 kinds of transfection reagents. And you can see that cell they can function normally here before and after using our products. So this is the SR buffer. And the third one is supplement, ITSM, and it is also stand as insulin, transferrin, and selenite. Those 3 components, they are the most essential growth promoting components for serum-free culture media, and they have a different functions for the serum-free culture media, such as insulin. It stimulates for the cell growth in the serum-free media, and transferrin is an iron carrier and also essential growth factors for the cells. And selenite is to optimize the cell growth and as an antioxidant. So these 3 components is very important and used a lot in the serum-free culture medium because they can sustain a good growth on the cells. And here is their concentration. So after that, we also test the ITSM on different cell lines, HAC 29030, HEC293 T cells, HEC293 U87G, and PC3 cells. So by the doubling time, you can see they responded to the ITSM difference, but actually they are growing normally. It is acceptable for their growing under this kind of condition. So we check their before and after culture. Their morphology seems normally as well, not so different. So after check their viability and their growth curve, we next move to their function test. So after do a transfection, the GFP proteins, you can see their positive GFP cells and rate is similar. So the ITSM can serve as good supplements to culture your cells in a low serum conditions. Okay. So this is some of the products we are serving for different using purpose. So among the previous contents that I introduced, I would like to summarize them within a takeaway message for you. So you can know that our Simply offers most commonly used cell culture supplies. So you can find the products about the cell culture media, valence salts, supplements, and antibiotics here. And if you have specific cell culture demands, you can consider our 1 million. It's a good alternative for major use special media. And also if your research is involving in reducing the FBS usage, you can consider our SR basal media, SR buffer, and ITSM. And if you have some customized demands to prepare your own liquid or powdered cell culture media, we can also tag this service as well. So the above is about my sections, the cell culture selections, and I hope it's clear for you, but if not, you are welcome to leave your questions and comments, then I will try to answer you. Okay, so I'm going to drop the presentation back to Jason.
All right, thank you so much, Harper, for sharing.
Thank you.
Like especially, I would say all of those slides that mention about the results when you actually incubate your cells, right, in those media, because that's important, isn't it? Mm-hmm. Right. So like we obviously need to know, right, when and whether like it's compatible as well. Because the last thing you do is that you have the media that doesn't match with the correct media, and after that you might have problems from there, right? Okay, and you never know until later on, isn't it? Okay, yeah. All right, thanks so much, Harper. Yeah, so I'll just continue on my side. Okay, now we'll give a bit of a break here just to show you that if you're interested to get the culture medias, all right, that Harper has shared, okay, even those additional supplements as well, do let us know. Okay, and I would actually now mention that we have another quick poll here as well. Okay, since we have mentioned a lot about the cell cultures, so we want to understand what kind of media you're using as well. Okay. Yeah, just a quick answer. One of the question came up in the chat, right? The medias are pharmaceutical grade as well. Okay. So I know some of you had the question, but yeah. So go ahead. You should see a poll on your screen. So asking which kind of media you're using. Okay. Because every single cell culture you're doing right now might have different kinds of media. Okay. Now, as you guys are filling up that, okay, I will mention that if you are looking for cell culture media and other balance cell solutions, or even some reagents or supplements as well, okay, do let us know. And if you say you just want to get some consumables, okay, we do have some from different brands, Flask, like T25, T75, T125, cell culture plates, clear, V bottom, U bottom, black, white, centrifuge tubes, okay, physiological pipettes, 5 mils, 10 mils, 25 mils, certain cell scrapers with different widths, okay, syringe filters as well, because I saw some of you might do MTT, and also culture dish as well. Okay, so do let us know, and do select which ones that you're looking at. Okay, we're not done yet, by the way. Yeah, so do stay with us. Okay, yeah. So just for time, I'll look at the responses right now. Okay, so far generally, we do have some that use culture media that is beyond whatever is listed here. Interesting. Do let us know as well, because sometimes we can work with Harper from Simply, okay, to formulate some others that is not listed over here as well. Okay, but I see most of you guys are doing DMEM and RPMI. Yes, these media are actually the most general ones that that most researchers are normally using as well. Okay. All right. Due to time, I'll move on from here and cover the last part for today. Okay, is obviously what can you do with it, right? You know all of the different techniques. You know the different cell lines. You know the contamination. You know that's supposed to be done in the lab. But if you don't know what is it done for, then it's kind of pointless, isn't it? Okay. So couple of applications here in categories here. This is the main ones that I normally see. The first one is normally on cancer research. So normally for this, they use cell culture to basically understand the interaction and the role of infection as well. Okay, so normally they will get insights into the biology of cancer, and they do through selective treatment of wild type cells. Okay, sometimes with UV radiation, viruses and toxins. Okay, or even certain causative agents as well. Okay, okay. Another application they normally use is for vaccines. And basically, this is where they would be using as host for viruses to replicate. Okay, so they want to study growth rates, development, conditions required for infectious cycle. So diseases like polio, measles, chickenpox, rabies, hepatitis B, and you can even put COVID as well, right? Okay, we're all done in here. Okay, using at least a cell culture as a model or host. Gene therapy wise. Okay, gene therapy wise here, okay, is more to help to express specific genes to see impact when you put new DNA or RNA into the system, and you want to see whether any specific pathway is influenced by them or even silenced by them as well. Okay, now next one about recombinant proteins. Okay, so this is they might use cell culture to actually produce a lot of proteins. Okay, and also to see how particular proteins regulate biology as well. Okay. Drug development wise is more on basically screening novel chemicals, cosmetics, drugs, and compounds, okay, and to see any cytotoxicity as well, okay. Also, you want to see whether there's any possible screening for target specific types at certain doses, and you also want to figure out what kind of dose that is non-toxic as well. So I can share, excuse me, like my zinc oxide nanoparticles work, right? I was trying to identify between 5, 10, 15, 20, 25, up to 50. So I found that it's around 20 micromolar, right? So that's through a couple of rounds of treatments and replicates, and I know, okay, that's the amount that it doesn't kill. Because think about it, the zinc nanoparticles that I was working with is good against UV, so for sunscreens, yeah? One example for drug selection. Okay, now also stem cells is used because you want to differentiate the specific or specialized cell types. Okay, and normally done in 3D protein matrix. Yeah, and lastly is IVF. So if you have a sperm cell and ovum cells, you want to help women to get pregnant. So to treat infertility or some that have genetic problems. Yeah. Okay. All right. What I'm seeing so far is that they are moving towards hydrogels. So it's currently one of the very more popular cell culture methods. Okay, to do so. Okay, so some of the examples here is 3D culturing here, 2D hydrogel, static suspension, okay, hydrogel cell bead, and even for in vivo studies where they create the cell in 3D form and then inject it. Okay, so these are the 5 common types I normally see. You might be asking, why? Why 3D? Okay, so actually, right, think about it. The cells in our body is not flat and squished onto 2D, as you can see on the left here. Okay, cells are all mostly in a 3D matrix with our tissues because we ourselves are also in 3D. We are not flat. Okay, so by having cells in a hydrogel which replicates closely the 3D matrix, you're actually allowing the cells to mimic the natural environment or where they are. Okay, so like From there also, because different parts of our body has certain rigidity or stiffness, you can actually adjust that to match with the natural stiffness of where the cells come from, like between liver or lung or brain. Because some of them are not like hard as bone, and some of them are not also as soft as tofu, for example. Okay, all right. And unlike the 2D culture, which I think you just now you saw me explain about the methods, okay, the flow. You don't need that complicated one, okay. So you don't need sophisticated protocols. You don't need certain materials or devices to grow. You just have to put it onto a gel, and you should grow by itself. Yeah, okay. And I'll finish off with the last slide here, basically mentioning, with that in mind also, there are different types of cell based applications matching with the 3D. Okay, so you got stem cells, okay, and you got neural cells, cardiac cells. Okay, so basically you can see that stem cells are more for drug screening. Neural cells is just to see functional network, and then cardiac cells is for heart related. Okay, you got liver ones as well, toxicity. Okay, cancer cells especially to see whether there's any changes. Okay, and also some others as well. So whether you're doing any tissue engineering, cell differentiation, so on. Okay, so I would say very, very big thank you so much for staying. Okay, all the way to the end here. We got still a few more things like Q&As and so on. So you can please follow us by all these social media sites and including our website here. And I'll pass back the floor to Vicky. Vicky, go ahead. Vicky, I think you're muted.
Yes. Okay. Thank you so much, Dr. Tan and Ms. Harper, for the extraordinary presentation. It was nothing short incredibly informative and interesting. So before we proceed to the Q&A section, I would like to grab back your attention to the screen for our in-webinar only exclusive offer. Okay, get your 10% off for any Simply cell culture media and reagents with no minimum orders. Be sure to place your order by 30th April to entitle for this promotion. So claim your voucher now in the chat box. Okay, so before we go ahead to our questions, I would like to urge all of you to turn on your camera for our photo session.
Don't be shy, come.
Yeah, okay, so please hold your smile for maybe 10 seconds and we will take a screenshot for it. Okay.
I really highly appreciate you guys actually staying this long, yeah? We got a lot of things to cover and wanted to make sure that you guys got information worth the time, yeah? Okay?
Yeah, just hold your smile for 10 seconds and when it's done, I will inform you all. Okay, thank you so much for your cooperation. So now we will moving on into our Q&A section, and for this you all actually can already leave a presentation. We actually will share the slide, the recording video to you after we end it through email. Okay, so wait lah. Sorry. Okay, so the questions that has been answered in the chat, I will not repeat it. I will just read out the questions that have not been answered. Okay. So wait for me to scroll back. Okay, so the first questions from Claderon. Okay, when Dr. Tan, when doing a passage in the confluency observed in the microscope, enough to change the media? Is it always necessary to count cells in the hemacytometer or just watch the span media and confluency?
Okay, you watch it will do. Because if you start to disassociate, right, then you would basically add another passage, and if you do that, right, then your results might differ. Okay, so normally observe first, and then when you feel is ready, this is based on experience. When you feel it's enough, then only you perform the counting. Okay, yeah.
Okay,
That's my answer.
so next question, should all caps place upside up or upside down in the laminar flow cabinet during handling?
As I said earlier, right, it depends. I personally have put it up because when I put it down, if you think about it, right, the surface might be dirty. So I put it up so that I don't have anything on the cap as well. So I would say play by ear on this one. I've read some say put down, some say put up. I would say go with what your seniors have done. So if they say up, you do up, down, you do down. Obviously, for culture plates, you put up, of course. Okay.
Okay, for next question from Chong Pan Pan, should we use filter tips for cell culture work? Is it necessary?
No, most importantly is whether your tips is sterile. Okay, filter tips is more on like, okay, I know where you're coming from. Two things here. First is that your tips itself, make sure it's sterile. Don't care whether it's filtered or not filtered. Okay, the filter part is more on if your pipette is dirty, then yes, you can use filtered tips. But if I'm not mistaken, you can't autoclave filtered tips. So you got to think about that as well. So if you want to get filtered tips, you need to get it always in racks, which might be more costly than actually getting non-filtered tips in bags, put it into a reusable rack and autoclave it. So you got to think about that as well. If you have the cost, yeah, by all means go for filtered tips, right? If not, then even regular ones is fine. Okay.
Okay, so the next question by Mohammad Fahmi. The liquid nitrogen space is limited in my lab. Can I just use negative 80 degrees Celsius freezer, I suppose, in my work?
Yes, I have used, I've like put the cells in negative 80 for about 6 months to a year. So yes, you can do so. If you're keeping it much longer than that, then I advise to put it, don't have to be minus 80. I've also put it minus 150 as well. So it depends on what you have, but as long as it's negative 80 and below, and you're going to use it for your duration of your experiment, and cross fingers that nothing happens to your fridge or freezer, then you're good. Okay.
Okay. Okay. Next questions by Elaine. Dr. Tan, could you elaborate further on the effects of the pipetting, comparing holding straight and upright to aspirate the sample versus holding the pipette slanting sideways at an angle? Which is better to get the accurate volume? Thanks.
Okay, so this one, if I'm not, might have to get back to you by this on email. But as far as I understand right now, right, having it in slightly an angle is a little bit better than having it upright. Think about it. If you have it upright, right, this is a bit on physics. If you have it upright, you're going against gravity, which is a lot of effort. If you put it towards the side, you have less gravity affecting it, and therefore you're able to aspirate a little bit better. And again, when you are using regular pipette, right, it's all basically playing with vacuum, right? So when you are pulling it up, you're creating a vacuum, and therefore you're allowing the media to come up. So it's actually, technically speaking, easier to slant it slightly and actually do it. Okay. Speaking about that, right? We do have pipettes, right? From Vista Lab, right? That is angled in 45 degrees. So it's already what they call solving that issue that I was mentioning about the gravity. Okay. Yeah.
Yep. And for the Vista Lab pipette, right, is if you require any demo, kindly contact us. We have the demo unit with us as well. Okay, so next question from Natasha. Is hitting the media few times is bad too? Your
Okay. Basically, no. As long as you don't hit it beyond 37, it's fine. Because think about it, your cells are already in 37 degrees anyway. So your media is going to be in 37 degrees, right? Most of the enzymes in there and the proteins doesn't would only deformed above 60 degrees. Okay, so if you put it beyond 37 or so, a little bit over, it's fine. Most importantly is that you only warm up your media when you're going to use. So you also reduce chances of some of the nutrients in there from degrading. Okay, so what I would normally do is that I would have a main bottle. I'll take out and put into a small 50 ml tube that I'm going to constantly use, and I'm going to warm that only. So you are reducing chances of your main bottle from degrading in terms of the quality. Okay, that's my suggestion there.
Okay. Another question from Haslinda. Some colleague told me that mycoplasma can also be detected by DAPI staining, DAPI staining. Is that right?
Okay, so you want to understand that I think for DAPI and host, right, you're trying to stain the nucleus. So bacteria got nucleus also, right? So bacteria is small. So theoretically speaking, you could, because DAPI is for the nucleus. So yeah, you could, but you can just double check that online as well. Yeah, just to make sure. If there are protocols, then yes. Okay.
Okay. Next question from Liu Fei Hong. For the contamination, endotoxin contamination, any precautions measure?
Well, you can get an endotoxin removal agents. That would be the best. Okay. I think that would be the only answer I have for endotoxins. So obviously, other than the aseptic techniques that you have, okay, and making sure that you're working in a lab that's away from bacterial contamination or sources, so that you avoid endotoxins. Okay, because that one is more on the chemical side of things. So like chemically affecting your cells. Okay, we do have some solutions if you need for endotoxin. Let us know as well. Yeah.
Okay, so another question from Liu Fei Hong as well. Hi, kindly clarify if we assign third party to perform microbial contamination tests and mycoplasma tests on our cell culture, do we send the sample in cell form or spam media is good enough?
The last one, what you mentioned again, cell form or
Spam media.
spam media. Okay, I'm not sure what you're talking about, but the last bit, but okay, if I understand correctly, right, I think what you're saying is that you're trying to send the media, okay, your cells either in the flask over to do testing. The question I have here is that your cells, right, supposed to be incubated. So I don't think you could send a cell that has already been cultured or currently in the incubator. If you say sending a passage, right, to check, then yes. You say sending media to send to check, then yes. Most of the time, right, I think they should be coming over to your lab and do the test there. They should do a very quick one there to check. That's my answer for that. Yeah.
Okay, another question from Lunazia. Does Gaia size supply culture medium for anaerobic bacteria?
Oh, that is beyond this scope of this because that is related to microbial, which I've done a webinar in November. You can check that. You can get a link from us for that to watch that webinar. But yes, we should have for anaerobic bacteria for culture media. It will be from Huang Kai. Okay. Yeah. I think this one you can email to us. We can take it later. Yeah.
Yep. Okay. Another questions from Li Xiaowei, sorry. May I ask, is THP1 macrophage a continuous or finite cell line?
Oh, this one I don't know out of my head. I have to reply you by email and check for you. Yeah. Because there are many cell lines out there. Some are finite, some are continuous. But I can assume here is most likely a continuous because it's a cell line. Right. Normally there's a specific name that has been publicized many times. They have mostly been sourced back in the 50s, 60s, or 70s, or even 80s, right? From the same cell line, and then they just basically repackage it and sell you the different passage. Right. But best is to check it online on this one. Yeah.
Okay. Questions from Liu Fei Hong. Is the primary culture similar as cell explant?
This one I need to take it offline as well. I will try and answer it after this. Yeah.
Okay, so questions from Muhammad Fahmi. I have no experience in cell culture, but will be using microphage primary cell line. Can I detect cytokine in this cell culture after infecting with human cytomegalovirus?
Okay, I think you could by ELISA kits. If you know specific cytokines, you can use ELISA straight away to detect. Okay, but I think you need to perform some extraction to do so. Okay, yes, macrophage cytokines, actually most of the other cell lines also do have cytokines as well. So if you're wondering, yes, you could do so. Yeah, ELISA would be the one I would recommend straight away. Yeah, I'll leave it there.
Okay, question from Rashichin Rasli. Is the way of clean or decontaminate the lab or incubator different if the contamination is from different type of contaminant, such as bacteria, fungus, yeast, virus, or other contaminant?
Yes. So you're saying, sorry, can you repeat the question again? Just want to make sure that I hear it correctly.
Okay, so the way of clean or decontaminate the lab and the incubator, is it different if the type of contaminant is different also, such as
Oh, okay.
bacteria, fungus?
I understand now. So, no, because right, if you perform decon, right, okay, sterilization, right, it affects all microbes. Don't care whether bacteria, yeast, and so on. I know what you mean, because you might be saying bacteria, yeast, mycoplasma, so all might grow differently. Okay, but no, if you do decontam, right, above a certain temperature, right, which is normally done about 120 to 140 degrees. Don't care whether it's a new air brand or other brands, right? Most to all microbes, even normal cells that you have also would be killed already. Okay, so yes, I would say for most cases, the typical ones that you normally do to clean, even ethanol and also decontamination, fumigation as well, it would definitely kill most to all of it. And you can always ask them to do it a bit longer, right? Just to make sure that you have that consistency there, and then like you're able to like keep the heat there for a while to keep the fumes there long enough so that almost to all microbes would go. Okay, yes.
Okay, questions from Muhammad Rasdan Hadi is quite technical and specific. For anti-fertility testing using plant extract, would studying on 15P1 Sertolic cell line be sufficient, or should I include other cell line too, like BALB, 3T3 fibroblast cell line for cytotoxicity evaluation?
Okay. So can you repeat the first part you saying? I just want to double
Okay,
check.
anti-fertility testing using plant extract, would studying on 15P-1 Sertolic cell line be sufficient?
Okay, okay. This one it depends on your direction you're going. Okay, I will share my experience here. So initially, for me was to actually do like zinc alternate particles, and then after that against cerium, and then also against the you can say against, okay, basically it's zinc oxide particles and also cerium oxide particles, right? So I was starting with neuronal cells, and if that one worked very well, I could go to try different neuronal cells or different cell lines as well, right? So it'll be similar to yours where you say I have a plant extract and I want to see how well it would actually handle like in terms of cytotoxicity or even like other factors between different cell lines as well. So that's one direction you can go. Okay, this is more like broadening. Okay, that's one direction. Second direction is that with the current cell line you have, right, let me check other things. Let me check the pathway you're looking for, any proteins that upregulated, downregulated. If you want to go along the cytotoxicity route, you know, whether is it early apoptosis, late apoptosis, autophagy, necrosis as well, then it's going deep down. Okay, so I wouldn't say there's a single answer for this. It depends on you and depends on whether you have the access capabilities and also obviously the funding, right? To have many cell lines or to go one cell line and then maybe to do proteomics as well, which I did for my own research.
Okay, question from Fora Ahmad. Do post-treatment after contamination happen can affect the growth rate of the cell? And also, if we change different type of media to grow the cell, can it also affect the cell growth?
Yes and yes. I'll put it as that for you. So obviously, changing, getting contamination post is the same as getting pre. So there's no difference there. And changing the media, yes as well, because media, as Harper was saying also, right, has different nutrients in there as well. Okay, and it will affect slightly the cells. Okay.
Okay. Another question. Hello, does Gaia Science have a cell culture service for osteogenesis and angiogenesis tests if we send samples?
No, because we don't have a lab at the moment.
Yes.
Yeah.
Okay. Question from Tan Kwan Chuan. If we source the cell line from a third party, what documentation that we can ask to prove that the cell fits for our usage?
That is a very big open-ended question. Firstly, is that whether the cell line itself. Okay, so if you think third-party cell lines, right? Assume best case scenario is that it will work normally with any other publications, because normally publications will say follow ATCC, then you buy ATCC cell line, Merck cell line, Merck cell line. Okay, so if I put it this way, if it's third party that is not published before, or at least like there's some resemblance, it should work the same. Okay, so and then the only way to really know is actually you buy it and then actually you basically revive it and see how it performs. If it performs based on some of the publications that you have read and it does the same thing, then it's good. Okay, there isn't really like a documentation prior to that to really tell unless there's publications. Okay, yeah, that's my answer for that.
Okay. Questions from Liu Fei Hong. If we perform in-house microbial contamination tests towards our cell in the laboratory, do we still need to prepare positive control and negative control for the test? Concern is if we prepare positive control, it will tend to contaminate our laboratories of cell culture.
Yeah, I see where you're coming from, because yes, if you try to have, okay, best way is you have two separate incubators, right? So you have one that is bacterial one is not. Okay. I haven't personally done that kind of approach, but I know where you're coming from. So this one you can trial error on your own. So you're saying one is positive and one is not. Yeah. So the thing is that if you're going to do that in the same culture hood, you might cross contam. Right. So yeah, I would say that in this case you try to separate it as much as possible. So you might have to do it on a maybe what you could do is that from the same one flask is split into two, and after that you have one is positive and one is how you will normally do it. And then after that, when you try to detect it, then you try to separate it as much as possible, so that and you need to be also alert, right, on which incubator you're using for positive and not, because those that positive one you definitely need to decontam after that, the incubator. So it's a lot of process involved if you wanted to do that kind of testing. That's why I was saying is that just do what they call some of the microbial testing I was mentioning, or even just put pen strap straight away. Then you should solve most of the problem for a short to medium period of time, if especially there's already SOPs for that. Yeah.
Okay. Question from Noor Shafika. What is the difference between acutase and trypsin?
Okay, so I was actually forgot to mention that. So there, in terms of I personally use acutase as well. There is what to say? In terms of dissociation, there's trypsin, EDTA, tripol, and acutase. So I believe it's just the enzymes that it's involved. And as far as I know, acutase is the easiest one, right? I was using my neuronal cells, right? And within 5 minutes, without knocking, put in incubator, all already disassociated and they're still separated as well. Trypsin that I used, it was still sticking after half an hour. But acutase is the most expensive one out there. So you got to play by ear. My suggestion, if you're trying to work on which one to work for, try with trypsin first. Then if it doesn't work, go to trypsin DTA. It doesn't work, go to ours is try TRYP-RC, and then after that, acutase. If you have the money, go straight to acutase, right? And then don't worry about it. Okay, yeah.
Okay, so actually, I think we will be ending our webinar on 12:30. So we will try to answer maybe 2 or 3 more questions. So for those questions that we are not able to answer, we will drop you an email for your questions, to answer your questions. So let's go for some short one. Note from North Africa, do we really have to deactivate our serum preheated with high temperature, bracket?
Deactivate serum. You have to understand what's the purpose of it, because normally high temperature for serum. I'm not sure about. Maybe you need to clarify with us what is the reason. So, yeah.
Okay.
I'll leave it as that.
Okay. Questions from Sim Sang Sai Peng. Certain multimode reader allows us to do drug treatments within the reader that has temperature control and CO2. Reading can be taken at multiple time points without us taking the plates in and out of the readers and the CO2 incubator. Would this affect the osmotic pressure, say a 100 microliter volume for 24 hours?
Okay, I know what you mean. So I have done the other version you mentioned. Put in, take out, put in, take out, put in, take out. Yes, it would definitely affect. This is why that there are solutions out there right now, even with our CytoSmart as well, right? The Omni, where you put it into the incubator, and then after that, if you're not doing any fluorescence based, okay, if you're doing just morphology detection, right, and also counting and so on, then having our Omni in the incubator with the culture plate, right, and obviously already temperature, CO2, and osmotic pressure, it would be balanced. Yes, those that you're mentioning, I'm not sure which brain you're talking about, but yes, if you put it temperature, CO2, long term there, definitely you will see evaporation. So this is something that you need to think about as well when you're doing it. Normally, that's why I put in and out a couple of times for that reason. There should be a solution for osmotic pressure if they're doing temperature and CO2 already. Yep.
Okay. Next question from Juliana. I think it would be directed to Ms. Harper. Peripheral blood mononuclear cells growth in DMEM? Questions mark. Should I supplement the media?
About this question, I think it needs to be checked whether where the blood mononuclear cells you get from, and you can basically, if you are not sure, maybe you can check on the publication about users they are doing this kind of experiments. Then you can check their culture conditions, so you know how to prepare your cell culture medium. So basically, it needs to be added some supplements, but maybe from the publication will be more accurate.
Okay. Another question from Liu Fei Hong again. Hi, Ms. Harper. Is the culture media require cold chain storage and transportation?
For the cell culture media, because basically it needed to be stored at 4 degrees Celsius, but during transportation, it can be allowed like 1 degree lower. So under room temperature shipping, it should be all right, just not the weather not be too hot. It should be okay. But basically, once you've already received it, just put it back to the refrigerator, will be fine.
Okay. Questions from JB Lab. Morning, is there difference when ATCC number difference but same strain? For example, like Salmonella typhimurium has ATCC 13311 and ATCC 14028.
About this, I think probably this should be the difference from the two colonies they harvest. So if they should be on, maybe they harvest from the same origin, but because they are cultured it in dependently, because they need to separate the cells and grow it separately to make sure, because the two clones probably they have the genetic difference, so they will name the items separately. So maybe they are from the same origin, but probably their genetic expression might be not all the same. Yeah, that's a difference.
Okay. Questions from Jenna Yusuf. Is the media GMP graded?
We have two lines for manufacture the cell culture media. One is GMP line, but you know the GMP line maintenance is very expensive. So if you need the GMP grade cell culture media, you need to by request. But normally the product we offer is for research use only, so it's not GMP grade. But we have the facility for GMP grade culture media manufacture. Yeah.
Okay, so okay, another question for Dr. Tan. For the darkened autoclave tab, is it an indicator that the item has been autoclaved?
Yeah, of course. That's all right. When the tape, so if you get the tape right, it's very faint, the strips. Then when you put it through autoclave, right, which goes high temperature, then it becomes darkened. Then you know that's autoclave. So it's an easy way to know whether if you put it into the incubator, it worked or not, right? Because sometimes it might also fail. Okay. Yeah. And this is something you also can check whether your item can be taken in the biosafety cabinet or not as well. Okay.
Okay, I think we will go for another two questions. So the question is from Noiza. If I don't have liquid nitrogen, how long can I keep my cell in -80 freezer for cryopreservation?
Okay, I can say from personal experience that I've kept it like as long as possible. So I think about a year or 2 years. Okay, it depends on how well and how confluent as well you kept it as well. If you preserve it, that's more important than actually not just the temperature. Okay, so yeah, I would say a year to 2 years is good. But just make sure that you do plan to use it like within a year or two. Even keep it much more longer, like a couple of years, then best to go below 80. I would say the colder it is, the longer it is. Okay?
Okay, last question from Francis Ting. Any risk of contamination from storage in liquid nitrogen? How to prevent it?
That's a very good question. Liquid nitrogen. Okay, so unless the liquid nitrogen is contaminated, right, or you did not close it properly, right, then it's possible. That's the only way I can think of, because even the bacteria itself would also be, all the bacterial contamination also would be preserved, right, in the liquid nitrogen. So you only would know when you actually thaw it. Okay, that's my answer.
Okay, I think that's it for the last questions. For others unanswered questions, we will reply you personally through email. So before we end our event today, would like to invite Dr. Tan and Ms. Harper for any last word for the participant.
Harper, go ahead first.
Sorry, I just missed.
Any last word for participant? Do you have anything to say before we end it?
Okay, thanks for your time. And if you have any question, you can contact us through our info email or contact Gaia Science. We are happy to serve you. Thank you for your time. Thank you. Thank you, Jason and Vicky.
Well, I would just end it basically saying that, cell culture would be if you're doing it, will be probably a few years of your life. So take it seriously, but also have fun doing cell culture work as well. Okay? Yep.
Okay, thanks. We would like to take this opportunity to thank our speaker, Dr. Tan and Ms. Harper, for the presentation, and not to forget all our participants today. So we hope you enjoy it and have gained much knowledge. So we will end our session here. Thank you, everyone. You may leave the webinar now. Thank you.